Okay, so our next speaker is Dr. Jean Van Buren. And Jean is, let's see if we can find your presentation. Jean's a former trainee of this program, but she's since moved on.
I'm coming full circle now.
At EPA in Cincinnati. So we've enjoyed having the opportunity to work with Jean.
Thanks, David. So I got a really good introduction there to most of the analytical methods for PFAS that I'm talking about today. I come from EPA's Office of Research and Development. We are non-regulatory. We're basically the R&D branch of EPA, and we don't have to worry about enforcement, compliance, things like that. We can just do research. So most of that research involves developing methods and then applying those methods, doing large-scale field studies. So this can involve monitoring or more treatment-related studies. But when it comes to PFAS, where EPA is at, we're kind of at the end of phase 1, where we've put out a bunch of targeted methods. The last ones, 1633 and 1621, were finalized in January of this year. And now we're looking forward into a future, focusing more on precursor compounds and this idea of a total PFAS method, which, as David just said, doesn't exist, and it's pretty much impossible. But it's going to look like a series of methods run in combination with each other to fully characterize PFAS in a sample. So the first method I'm going to talk about, 1633, is now kind of the go-to method. It's taken over because it is the most comprehensive method. It's a targeted method. It's reproducible. Use it for quantification, and it covers a wide variety of matrices. It's not a drinking water method. It's pretty much all other matrices. But there are some limitations, even though the target list now covers 40 compounds. So it's our most extensive method. 40 is not enough, definitely not enough, especially when it comes to AFFF. This method also covers solids like soils and sediments, but it has some downsides when it comes to things like biotissues, especially fish tissue. 1633 is particularly susceptible to matrix interferences, and PFAS has similar mass transitions to a lot of organic acids, fatty acids present in biological material and tissues that you would be using this method to look for PFAS in. So the second method that or the first method I'm going to talk about today that we're currently working on and developing is top assay, which David just introduced. So the reason why this is so hard to standardize is because it's difficult to make it reproducible. And if you can't make it reproducible, it's not defensible. The top assay is basically an oxidation step followed by just an analytical method like 1633 that you can add on. But the oxidation step has some downsides. It can, you can have a really simple sample matrix like groundwater or drinking water, where there's not a lot of other kind of scavengers for the oxidant, but then you can have something like wastewater, where you pretty much used up all of your oxidant and you haven't fully oxidized PFAS. So one of the ways we're addressing this is the use of that oxidation surrogate, something to tell you when your reaction is truly stopped. The commercial labs who are running these methods right now, we've been interviewing them along the way. They are aware of this, and they will sometimes go back and re-oxidize a sample over and over and over again, adding to the cost every time. PFAS analysis is really expensive right now. It can cost, $300 a sample in best case scenario, maybe $1,000 worst case scenario. So if you have to do 5 rounds of oxidation, that just adds to your costs. So the goal with this method is to publish a water method. It's probably going to be a non-potable waters like groundwater, surface water, wastewater effluent. We can add that in. And then we will evaluate whether we can do a solids method, so a soil sediment type method, especially useful for AFFF contaminated soils. And this is an effort that is that only is possible with a bunch of different collaborators. So we're talking to the labs who develop the top assay, the people who've been using it for years and years now, like the commercial labs, other people who've done some research into alternative methods for the top assay to see what is the consensus coming out of the research community of how do we actually make this reproducible. I've been told that it takes 5 years and $3 million to develop a standard method, and we don't have either of those things here. We're trying to do it at maybe not even a tenth, maybe less of that time and that cost. So we'll see how it goes. We're still, evaluating whether it is possible, but it's looking good. Solid waste, things like that. So we're adding top assay into this category of screening class-based methods that complement the targeted methods that are already in SW 46. And then I'm moving on to an example of an application of the top assay. Now, this is a project going on. It's a large wastewater monitoring project that we are using top assay in a suite of other complementary methods. But basically, this project kind of spun out of the idea, evidence popping up that PFAS in wastewater is extremely variable. And there's evidence that it can vary on an hourly basis. So how do you take a grab sample from a wastewater treatment plant and then use that one single number for the rest of the year when you're talking about that PFAS at that facility? That's probably just not possible. So we're starting, we're wrapping up some of the facilities that first came online in the study now, but it was a year-long monitoring project of biweekly sampling at 6 different wastewater treatment plants across the country. Different sizes, different treatment processes, and we sampled at every single point in the treatment process as well, trying to get a picture of not just what does PFAS look like coming into the plant and what does it look like going out in the residuals or the effluent, but also what happens at every step in the treatment process. Where does it partition, and does PFAS get removed at any point, or does it increase? Does it get added, mostly by the transformation of precursors into terminal products? The part 2 goal of this project will be identify those points in the wastewater treatment train or the cycle of residuals coming out and then going back to the front end of the plant, either directly or indirectly, where you can target PFAS removal and destruction at the point at which it's most concentrated and easy to target. So here's an example from one facility that separates their industrial wastewater from their municipal wastewater. These are different sampling events over the course of the year. The pre-top assay sample is basically the influent, PFAS and the influent coming into the plant. And then post is post-top assay. So after that sample has been oxidized, and does PFAS increase or decrease after that oxidation? So in the red here, we can see an example where 1633, used as the kind of targeted analytical follow-up to top assay, isn't good enough, and we're missing things. So we've oxidized either regular PFAS that we're already looking at or precursors into things that we are not measuring through 1633, and that's likely the ultra-short chain PFAS. And then the blue here, this is kind of the reason why top assay is so powerful, where we are producing more terminal PFAS after oxidation, and we know that that's coming from precursors that were not included in the analyte list in the beginning. Then purple here is one of the reasons why we would need to use an oxidation surrogate in the first place. So this lab has been running a top assay for quite some time, and they only do one step of oxidation. They don't go back and reoxidize this sample. So their one step of oxidation on that particular day for that particular sample wasn't enough to fully oxidize the precursors. This plant also has a really unique treatment step that's basically a low pressure oxidation step. And the plant actually sees increases in PFAS after they do this treatment. And that's because they are truly converting those precursors, which didn't show up, into terminal products. This is an interesting case scenario. This is a pretty unique treatment. Not all plants, not many plants are doing this type of treatment at all. But it's a good example of what happens to PFAS when you try to treat your waste for other things. In this case, they didn't introduce this treatment step for PFAS, but this is happening accidentally. So here, top assay on the same samples before it goes into this low pressure oxidation step, we can't actually recreate what we see through the treatment plant. So we know that we're not exactly chemically oxidizing through the top assay what that treatment plant is doing. But this project, this wastewater project, is coming to an end now. We also have a bunch of TOF data, a lot more targeted data, and we are following it up with some non-target analysis. And that will provide us a larger picture of what exactly are these missing gaps that top assay pointed out. So I'm going to move on to a second application now. So this is a project led by Harry Beller over here, EPA Region 9 office. So this is the use of top assay in a different context. It's kind of looking at or addressing this research question of if top assay is basically an assessment of reactivity of a PFAS mixture, does that have any similarity with toxicity and reactivity of that same PFAS mixture biologically? So in this case, we are specifically looking at AFFF. We know that AFFF contaminated sites can be pretty well characterized just by the targeted methods and by top assay. We want to know what would the toxicity of that groundwater look like. That groundwater is difficult to remediate. It's being used for drinking water, for irrigation, crops and animals and people. All of that AFFF related PFAS ending up in the soil if it's used for crop irrigation. This is making it difficult to assess risk when we only know so much about PFOA, PFAS, and a small handful of compounds. If it took us this long to characterize their toxicity, we can't do that for mixtures. We need a better way. So one of these shortcuts is looking at this question of using the top assay actually as a surrogate for toxicity. So if you have precursors present, high reactivity with the top assay, does that actually translate over into the same thing on the toxicity side? So we are comparing AFFF mixtures with different bioassays to see if any of them correlate to top results. This includes cell painting, PPAR, alpha receptors. And then once we have a picture of how PFAS mixtures look with those different bioassays, then moving on to a second step of whether metabolism and oxidation on the same sample, on the same PFAS mixture, mimics what's happening with the top assay. And what does toxicity look like after a sample has gone through like a liver cell oxidation versus a top assay oxidation. All right, so then finally, I'm going to talk about the last method that we have in development. And this is just a non-target analysis as a general term. And you might laugh if you know anything about NTA and I say, we want to standardize it, because that's also an impossible concept. We will never standardize NTA. But we can possibly improve transparency and reproducibility. And EPA, especially ORD, is working on a bunch of tools right now that are helping in this realm, specifically the QNTA framework for semi-quantitative NTA, the CompTox dashboard, which is public, and those will all tie into the EPA web app for NTA, which is not public yet, but they're taking beta testers if you're interested. And that will kind of allow us to have some way of basically importing your data into a website. This is your processed NTA data, and it will spit out a QA/QC form report, give you some compound identifications, and give you some kind of preliminary risk assessment based on toxicity thresholds pulled in from the other tools there. So that is necessary because, like this picture shows, all of these projects are building up like large amounts of samples. And the cool thing about NTA is you can run NTA on the sample extracts that you did your target analysis on. So for our wastewater monitoring project, all of these coolers here, these contain test tubes of extracts that are just sitting there waiting for somebody to have the time to go run NTA on. And that someone is me, and I don't have the time. So I want all of these tools to get used and to come into public eye and get feedback that makes them better so that I can start running these samples 1,000 samples at a time and have reproducible data at the end there. And that's pretty much it. Thank you, everyone.